Imagine the planet before memory, before bone or leaf or shell. A young Earth turning under a dimmer sun, its surface a restlessly shifting mosaic of black basaltic crust and glowing lakes of lava, its skies the colour of iron and ash, its air heavy with volcanic exhalations that would scald our lungs in a breath, and its seas, where they existed at all, not yet the deep blue familiar from orbit, but a hot, mineral-thick cauldron in which water and rock and atmosphere ceaselessly argued with one another in lightning, steam, and shock waves from unending impacts. In that alien landscape there was no eye to see, no fin to cut the waves, no root to probe the ground, and yet in those same hostile conditions sometime more than 3.5 billion years ago, the first fragile boundary between inert and alive was crossed, setting in motion a chain of transformations that would eventually give us coral reefs and crows, oak forests and octopuses, and a species capable of wondering how the whole improbable story began. To reach that threshold, we have to step even further back, to a universe in which not even a planet existed, when all the matter that would one day congeal into oceans and mountains and membranes was compressed into a hot, rapidly expanding plasma born about 13.8 billion years ago in an event we call the Big Bang, where, in the first minutes, only the simplest nuclei, hydrogen, helium, traces of lithium, could survive the searing density. Everything else that life would need, the carbon in your bones, the nitrogen in your DNA, the oxygen that rusts rocks and fills your blood, had to be forged later in the hearts of stars and in the cataclysmic deaths of some of those stars as supernovae, each explosion sending shockwaves of heavy elements into the interstellar medium, seeding great clouds with carbon-rich dust and icy grains, so that when, billions of years later, one such cloud in a quiet corner of the Milky Way began to collapse under its own gravity and form a new star encircled by a whirling disk of gas and rubble, the raw materials for chemistry far richer than hydrogen and helium were already in place. Within that swirling protoplanetary disk, amid collisions and mergers of icy bodies and silicate rocks, one clump of matter near a distance where water could exist as liquid grew heavy enough to pull itself into a sphere. That sphere, our Earth, did not arrive in peace but through violence, including a titanic late collision with a Mars-sized protoplanet often called Theia, an impact energetic enough to melt much of the crust and fling a spray of incandescent debris into orbit, out of which the Moon coalesced, its gravitational presence stabilizing Earth's axial tilt, and with it the rhythm of seasons that later life would learn to anticipate. For tens of millions of years afterward, Earth's surface remained a hellish Hadean realm, named for Hades, god of the underworld, where the crust was thin and often molten, comets and asteroids hammered the planet in a chaotic rain, and the early atmosphere, stripped and replenished by impacts and volcanic eruptions, likely held no free oxygen, but was thick with carbon dioxide, water vapour, hydrogen, sulphur compounds, perhaps methane and ammonia, a greenhouse cloak that trapped heat from a sun fainter than today and helped keep surface water, once it finally condensed from freezing solid. Geologists infer this early drama not from eyewitnesses, of course, but from relics hidden in ancient minerals, tiny, nearly indestructible zircons that crystallized more than 4.3 billion years ago and now lie embedded in younger rocks in places like the Jack Hills of Western Australia, their internal isotopic ratios whispering clues about the temperatures and the presence of liquid water when they formed. From them emerges a surprising picture. Even while the heavy bombardment of the young solar system still rattled planetary surfaces, Earth may already have possessed a stable crust and cool enough conditions for oceans, vast basins of water collecting in the deeper scars of the planet, rain pouring from relentlessly overcast skies to fill them, thunder echoing across half-formed continents, lightning stitching from cloud to sea in flickers that, to a chemist, carry the promise of new bonds being forced into existence. Although to that world of rock and ocean there was as yet no environment in the biological sense, only gradients of temperature and chemistry, patches of acidity and alkalinity, mineral surfaces waiting to be etched and coated by complex molecules not yet invented. If the rocks hint that Earth's surface calmed quickly, the oldest convincing traces of life show up almost as soon as the stage is habitable, which suggests that the transition from non-living chemistry to the simplest living systems might not have required unimaginable improbabilities, but could be the kind of process that, given the right conditions and enough time, emerges relatively quickly. In banded rocks of Greenland's Isua belt, more than 3.7 billion years old, scientists study carbon isotopic ratios skewed in a way that, on modern Earth, living organisms produce when they preferentially use the lighter isotope, and in still young sedimentary rocks of Western Australia, around 3.4 billion years old, we find fossilised stromatolites, layered mounds built by microbial mats trapping and binding sediments as they grow toward light, testifying that by then, not only had life appeared, it had aggregated into communities capable of reshaping their microenvironment. Yet to understand how humans even came to ask where such life began, we must leave the Hadean and Archean seas behind and leap forward to a time when small bands of Homo sapiens stared at stars and water and wondered who or what had made them. For most of human history, the origin of life was not a scientific question, but an episode in sacred narrative, and those narratives, far from being mere fables, reflected the best attempts of early civilizations to interpret the world with the tools available to them. In ancient Mesopotamia, scribes who etched the Enuma Elish into clay tablets around the late 2nd millennium BCE imagined the gods themselves emerging from a primordial, undifferentiated water, a mingling of salty Tiamat and fresh Apsu whose conflict and union birthed heaven, earth, and the divine assembly, a story in which watery chaos precedes ordered existence, much as geologists later would say oceans preceded continents. Along the Nile, Egyptian priests revered Nun, the boundless dark water that existed before creation, from which the first mound of land arose bearing Atum, the self-created deity who generated other gods and, in time, all living things, so that each sunrise reenacted the moment life's stage first surfaced from endless fluid. In the Rigveda of ancient India, hymns speak of a cosmic ocean and a golden embryo, Hiranyagarbha, floating upon it, while in early Chinese cosmogonies, misty chaos condenses and the giant Pangu carves sky and earth apart, his dying body giving rise to mountains, rivers, and living creatures, myths that differ in detail, yet often agree in linking water, darkness, and a transition to ordered, breathing life, an intuition oddly in tune with modern theories that emphasize aqueous chemistry. Gradually, in a few cultures, attempts to interpret nature began to include proposals that sidestepped gods as immediate agents, and among the ancient Greeks of the Ionian coast around the 6th century BCE, philosophers who walked the harbours of Miletus and gazed at a Mediterranean horizon tried to understand change without recourse to divine caprice. Thales of Miletus, according to later accounts, declared water to be the fundamental substance from which everything ultimately derived, an idea that resonates faintly with the observation that life as we know it is water-based. Though Thales had no microscope or periodic table to support his claim, his younger compatriot Anaximander imagined a primordial boundless effluent giving rise to hot and cold, wet and dry, and proposed, remarkably, that at some early time life arose in moisture warmed by the sun, with humans or their ancestors first appearing as fish-like creatures that later came onto land, a kind of speculative evolutionary story 2 millennia before Darwin. Empedocles of Acragas, in the 5th century BCE, suggested that the world had once been populated by monstrous, incomplete beings, arms without torsos, eyes without faces, that gradually assembled into functional creatures under a kind of crude natural selection in which only viable configuration survived, an idea Lucretius would popularize centuries later in his Latin poem De Rerum Natura, where he argued that the universe was made of indivisible atoms colliding and combining by chance, and that living beings, like everything else, originated without design or foresight, a position radical enough that the manuscript had to be rediscovered in a monastery library during the Renaissance to influence early modern thinkers. Yet even as some philosophers entertained naturalistic accounts, the most influential biological thinker of antiquity, Aristotle, looked at the world and saw something else, an ordered hierarchy of beings, each with a fixed essence, embedded in a cosmos that favoured purpose and form over chance. Within that framework, he concluded, after observing decaying matter and the sudden appearance of maggots or mice in stored grain, that certain simple animals could arise spontaneously from non-living material, a belief called spontaneous generation that fit comfortably with his theory of potentiality and actuality, because moist earth or rotting meat, in his view, contained the potential for life which, under appropriate conditions, could actualize itself without parents. He wrote of eels emerging from mud, fleas from putrefying substances, and it seemed to everyday experience that jars left open to the air-bred organisms all on their own, especially in a pre-germ theory world where no one could see microscopic eggs or bacteria. So for centuries the idea that life continuously appeared from non-life under ordinary earthly conditions felt as self-evident as sunrise, so much so that medieval scholars, both in the Islamic Golden Age and in Christian Europe, quoted or adapted Aristotle without strong objection on this point. When the Roman Empire fractured and Europe drifted into the political and economic fragmentation once called the Dark Ages, attention to natural philosophy never wholly disappeared, continuing in monasteries and in vibrant centres like Baghdad and Cordoba. But the framing of life's origin shifted more firmly under theological authority. Jews, Christians and Muslims read their scriptures as describing a definite beginning of life in a finite past, an act of creation by a single God. Yet even as they affirmed that initial divine act, many theologians and physicians retained a belief in spontaneous generation as a secondary ongoing process ordained by God, a way to reconcile biblical cosmogony with everyday observations of vermin arising in filth. In medieval bestiaries one can find the persistent legend of the barnacle goose, a bird said to grow from floating barnacles attached to driftwood, leading some clerics to debate whether it might be eaten on fast days because, arguably, it came from the sea rather than fleshly reproduction, a little window into a mindset in which boundaries between living and non-living, animal and mineral, were conceptually more porous. Islamic polymath al-Jahiz in the 9th century speculated about the struggle for existence and environmental adaptation among animals, while still accepting that simple life could appear from mud, and later, in the 13th century, Thomas Aquinas carefully threaded Aristotelian philosophy into Christian doctrine, accepting that God was the ultimate cause of life while allowing that he might have instituted natural processes, like spontaneous generation, that operate without further miraculous intervention, leaving untouched the deeper question of how the first living cell, as we understand it today, could have emerged from pure chemistry. The first cracks in spontaneous generation's authority began to widen with the advent of better instruments, and a new experimental ethos in early modern Europe, when curious artisans ground lenses into shapes that could magnify drops of pond water into teeming universes. In the mid-17th century, Italian physician Francesco Redi, skeptical of the notion that maggots simply condensed out of rotting meat, carried out a simple but decisive experiment in Florence by placing pieces of meat in a series of jars, some open, some sealed, some covered with fine gauze, and watched as maggots appeared only where flies could physically reach the flesh, their eggs invisible to the naked eye yet clearly required, showing that at least in the case of higher animals life came from life. Around the same time, Dutch draper Antony van Leeuwenhoek, armed with exquisite single-lens microscopes of his own design, peered at rainwater, dental plaque and scrapings from his own skin, discovering what he called animalcules, tiny, wriggling beings no philosopher had ever imagined, which plunged the question of spontaneous generation into murkier waters, because now there existed a vast, previously unseen domain of life whose reproductive habits were mysterious, and many naturalists thought it plausible that, while maggots required eggs, microscopic organisms might condense from infusions of hay or meat just by virtue of some vitalistic principle pervading air and matter. Through the 18th century, debate simmered as experimenters like John Needham and Lazzaro Spallanzani boiled broths and sealed them in flasks, with Needham claiming that even after brief boiling, new microorganisms appeared, proving spontaneous generation, while Spallanzani argued that more thorough boiling and tighter seals prevented any life from arising, suggesting contamination from existing germs. The argument exposed how fragile conclusions could be in an era before standard sterilization, or an understanding of heat-resistant bacterial spores, and observers interpreted similar outcomes through different philosophical lenses, some clinging to the idea of a vegetative force latent in matter, others moving toward a germ-based explanation. It was only in the 19th century when Louis Pasteur, motivated not only by pure curiosity but also by practical problems like spoiled wine, devised his now famous swan neck flask experiments, that the case against spontaneous generation in ordinary conditions became nearly impossible to dismiss. He boiled nutrient broths in flasks whose necks were drawn out into long curved tubes open to the air, allowing gases to exchange but preventing dust and microbes from falling directly in, and discovered that such broths remained sterile indefinitely, until the necks were broken or the flasks tilted so that liquid contacted the dust-laden bend, whereupon microbial growth blossomed, showing that the vital force was not a property of air itself but of living contaminants, and prompting Pasteur to declare in 1864 that omnicellular e cellular, every cell from a pre-existing cell, was the rule, apparently slamming shut the door through which new life might enter the world. Yet Pasteur's triumph raised a new and troubling question for the scientifically minded. If in the present day no life appears without parents, how did the very first cells come to be, and does denying spontaneous generation for modern conditions implicitly require a supernatural creation at the dawn of time? Charles Darwin, who transformed our understanding of the diversification of life with his theory of evolution by natural selection, was keenly aware of this deeper problem, but curiously cautious about tackling it in print, perhaps sensing the religious and philosophical explosives it contained, so that in On the Origin of Species, he sidestepped the literal origin of life and ended the book instead with a famous flourish about a creator breathing life into a few forms or one. However, in a private 1871 letter to his friend Joseph Hooker, he allowed himself a speculative aside, wondering whether life might first have arisen in a warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, where a protein compound could form chemically and then grow more complex, only to be immediately devoured if such a thing happened today by existing living organisms that now would devour such matter or absorb it, which would not have been the case before living creatures were formed, an image that prefigured in miniature the later idea of a prebiotic soup, yet remained unpublished during his lifetime, a secret note passed between friends rather than a public declaration. It would take the upheavals of the early 20th century, scientific and political, to transform Darwin's private speculation into a systematic hypothesis about how Earth's chemistry could, over immense spans of time, bootstrap itself into biology. In revolutionary Russia, a young biochemist named Alexander Oparin, working in a scientific culture shaped both by Marxist dialectical materialism and by a fervent desire to modernize, grappled with the problem of life's first appearance and, in a modest 1924 pamphlet later expanded into a book, argued that there was no sharp boundary between living and non-living, but rather a continuum of increasing complexity shaped by selection on chemical systems, and that Earth's early atmosphere, lacking oxygen and dominated instead by hydrogen-rich gases like methane and ammonia, would have favoured the spontaneous formation of organic molecules under the influence of sunlight and volcanic energy. Oparin described how such molecules could accumulate in ancient oceans to form a primordial soup, out of which microscopic droplets called concavates, tiny phase-separated blobs rich in macromolecules, might form, exhibiting rudimentary properties like selective absorption and internal chemistry, foreshadowing living cells even though they lacked true genes. Around the same time, largely independently in Britain, the polymath JBS, Haldane, a Marxist and adventurer who combined genetics, physiology and popular writing, proposed a strikingly similar scenario in a 1929 essay, suggesting that before life, Earth's seas would have been a hot, dilute broth of organic compounds from which the first self-replicating systems arose, and he coined the phrase prebiotic soup to emphasize that these processes occurred before biology rather than within it, so that by mid-century a broad conceptual framework existed. Life had originated via abiogenesis on a young, reducing Earth, where simple molecules assembled into more complex ones, and eventually into systems capable of heredity and metabolism. This framework, however, still lacked a crucial element: experimental confirmation that such synthesis of life's building blocks under plausible primordial conditions was even feasible, and it would be an American graduate student, Stanley Miller, under the guidance of Nobel laureate Harold Urey at the University of Chicago, who decided in the early 1950s to test Oparin and Haldan's vision with glassware, electrodes and patients. In a now iconic setup assembled on a lab bench, Miller filled one flask with a mixture of water to represent the ocean and connected it by glass tubing to another chamber containing gases thought to mimic the early atmosphere: methane, ammonia, hydrogen and additional water vapour, then applied heat to boil the ocean and drive vapours into the atmosphere, where he sent continuous electrical sparks to imitate lightning before allowing the gases to condense and drip back, completing a crude cycle. After days of humming and occasional smells of ozone, the once clear water grew cloudy and slightly pinkish, and chemical analysis revealed that a suite of organic compounds had formed spontaneously, including several amino acids such as glycine and alanine, the very monomers that, in living cells, chain together to make proteins. When Miller and Urey published their results in 1953, the same year Watson and Crick proposed the double helix structure of DNA, the scientific world and popular press seized on the contrast between the apparatus's simplicity and the profundity of its implications, seeing in its boiling flasks a miniature reenactment of Earth's own distant past, even though debates soon followed about whether the gas mixture used truly matched early atmospheric composition and about the stability of produced compounds in the face of ultraviolet light and continuous energy input. The Miller-Urey experiment, despite its limitations, opened a door for prebiotic chemistry, inspiring generations of chemists to test how far such processes might go and under what conditions. Some modified the gas mixture to better approximate what later geological evidence suggested, a more neutral atmosphere dominated by nitrogen and carbon dioxide rather than methane and ammonia, and found that lightning still generated organic molecules, though sometimes in different proportions. Others explored scenarios in which ultraviolet radiation from the young sun drove reactions in shallow ponds or on mineral surfaces, or in which molecules synthesized in the atmosphere rained into the ocean, only to be further altered by hydrothermal systems. Miller himself quietly stored dozens of vials from his original and subsequent experiments in freezers, and only after his death in 2007 did former student Jeffrey Bader and colleagues reanalyze some of them with more sensitive techniques, finding an even broader diversity of amino acids than Miller could detect in the 1950s, including unusual ones not used in modern biology, but demonstrating the generative richness of such simple setups. Yet as chemists pushed further, they confronted formidable challenges. Synthesizing amino acids was one thing, but forming long, sequence-specific proteins or informational polymers like RNA under plausible early Earth conditions was quite another, and the soup concept, while powerful, began to look too vague and chemically fragile to stand alone, prompting some to look not just to Earth, but outward, to space, for contributions to life's raw ingredients. The idea that life, or at least its seeds, might come from the heavens is older than modern chemistry, with the pre-Socratic philosopher Anaxagoras suggesting 2.5 millennia ago that seeds of life pervaded the cosmos and occasionally took root on suitable worlds. But in the 19th century, as the scale of the universe and the age of Earth stretched far beyond biblical chronologies, some scientists revisited a more restrained version of this idea, called panspermia, not to abolish abiogenesis altogether, but to relocate its defining moment elsewhere. Lord Kelvin and Hermann von Helmholtz speculated that life might have begun on some older, now dead world, and travelled to Earth on rocks ejected by impacts, surviving the cold of space and the fiery passage through another atmosphere, while in the early 20th century Swedish chemist Svante Arrhenius proposed that bacterial spores, if small and light enough, could be pushed between stars by radiation pressure, although later calculations of interstellar hazards made such journeys seem unlikely. Even as mainstream biology leaned toward terrestrial origins, clues from meteorites quietly accumulated. In 1969, a fireball streaked over Australia and dropped the Murchison meteorite, a carbonaceous chondrite, into a field near a small town, and when chemists cracked it open, they found more than 70 different amino acids within, many of them distinctly non-terrestrial in their proportions and isotopic signatures, along with other organic compounds, showing that complex organics form readily in cold interstellar or protoplanetary environments and can survive the plunge to a planet's surface, a finding later reinforced by spacecraft missions like ESA's Rosetta, which sampled the atmosphere of comet 67P01 Churyumov-Gerasimenko and detected a medley of organic molecules, and by NASA's Stardust mission, which captured dust from Comet Wild 2 containing glycine, the simplest amino acid. Such discoveries strengthened a nuanced picture. Even if life itself did not arrive fully formed on a meteorite, the early Earth was almost certainly showered with organic molecules synthesized in space, from simple hydrocarbons to amino acids and sugars, enriching any nascent prebiotic chemistry with a cosmically sourced pantry. Modern concepts of panspermia have accordingly shifted toward lithopanspermia, transfer of hardy microbes or spores between planets within the same solar system, rather than interstellar travel, and experiments on the International Space Station and other platforms have probed just how resilient life can be under extreme conditions. Bacteria like Deinococcus radiodurans, famed for surviving intense radiation and dehydrated tardigrades, with their remarkable ability to endure vacuum and temperature swings, have been exposed to space for months, some preserving viability when shielded by even thin layers of rock or simulated Martian soil, suggesting that microorganisms buried a few centimeters inside ejecta from large impacts might indeed endure the journey from Mars to Earth or vice versa. Yet panspermia, even in this modest form, only moves the question back a step, because if life came here from Mars, one must still ask how it arose there, and if from an even older world, how it formed there. So for most origin of life researchers, extraterrestrial organics and possible interplanetary transfer are seen as augmenting, not replacing, the need to explain how a planet's geology and chemistry, under certain regimes, can cross the threshold into self-sustaining, evolving systems, an explanation that has increasingly turned attention from open ocean soups to more specific dynamic environments such as hydrothermal vents and fluctuating pools. In 1977, when the research submersible Alvin descended nearly 2,500 meters down the Pacific off the Galapagos Islands, guided by faint clues from seismic surveys and the delicate tether of its mothership, the geologists and biologists inside expected to see barren lava flows and cold, dark sediment, but instead their portholes filled with an otherworldly glow from chimneys of black, mineral-rich water spewing from the seafloor like underwater smokestacks, surrounded by dense communities of organisms entirely unknown to science. Giant tube worms with blood-red plumes, white clams the size of dinner plates, ghostly crabs and swarms of shrimp, all thriving in a realm with no sunlight, supported by microbes that chemosynthesized organic matter from hydrogen sulfide and other chemicals emerging from the vents. This discovery shattered the long-held assumption that life everywhere ultimately depended on photosynthesis, and suggested that deep beneath the waves, Earth harbored a biosphere powered directly by the planet's internal heat and geochemistry, leading some researchers to argue that such settings could also have been the cradle of life itself. In particular, geochemist Michael Russell and later collaborators like William Martin proposed that not the dramatic black smokers seen by Alvin, with their acidic, metal-laden plumes, but rather gentler, alkaline hydrothermal vents, structures like the Lost City Field in the mid-Atlantic, where warm, hydrogen-rich fluids seep from towering carbonate chimneys, offered a natural laboratory in which steep gradients of temperature and chemistry could drive prebiotic reactions, with the labyrinthine micropores of the chimneys acting as both compartments and catalytic surfaces. These alkaline vents, formed where seawater percolates deep into the oceanic crust and undergoes serpentinization reactions with ultramafic rocks, emerge at the seafloor as fluids saturated with hydrogen and methane, sharply contrasting with the relatively oxidized carbon dioxide-bearing ocean water around them, so that at the interface, along mineral surfaces of iron-sulfur compounds and other catalysts, electrons tend to flow, offering an energetic push for reducing carbon and building more complex molecules. Russell and colleagues noticed that the scale and structure of the chimneys, honeycombs of tiny interconnected cavities bounded by thin inorganic walls, resemble in some ways the compartments within cells, and that across these walls, natural proton gradients can develop as alkaline vent fluids meet more acidic seawater, echoing, in a geochemical idiom, the proton gradients that living cells everywhere use to power vital work. Peter Mitchell in the 1960s had shown that cells rely on differences in proton concentration across membranes to synthesize ATP, the universal energy currency, via rotary molecular machines called ATP synthases, and decades later, biochemist Nick Lane and others seized on the similarity, arguing that life's universal dependence on chemiosmotic gradients may be a direct inheritance from an origin in such vent environments, where early protometabolisms harnessed pre-existing gradients before evolving lipid membranes and sophisticated protein pumps to recreate them in new contexts, ultimately freeing living systems from the stone womb of the vents. Despite the elegance of the hydrothermal vent scenario in solving the energy problem for the origin of life, how to drive uphill chemical reactions without modern enzymes, it raises vexing issues of its own, especially concerning the stability of delicate informational molecules like RNA at high temperatures and the difficulty of concentrating key ingredients in vast oceanic settings. In response, some researchers have shifted their gaze shoreward to environments where heating and cooling cycles, evaporation and refilling, and close contact with mineral surfaces could facilitate polymer formation and selection in ways less plausible in the deep sea. Biophysicist David Deamer, fascinated by membranes and the role of lipids in life, travelled to volcanic regions like Yellowstone National Park and New Zealand's Rotorua to study hot springs, whose fluctuating edges accumulate organic films as water levels rise and fall, and he proposed that on the early Earth, similar pools fed by volcanic gases and perhaps enriched by atmospheric or meteoritic organics would experience regular wet-dry cycles that could drive condensation reactions, linking small molecules into longer polymers as water evaporated and then testing their stability when the pools refilled. Laboratory experiments simulating such conditions, with thin films of amino acids or nucleotides drying on mineral surfaces or within microscale cracks, show that surprisingly long chains can form without enzymes, especially when aided by activating agents or by simple physical processes like capillary flow, and that amphiphilic molecules, those with both water-loving and water-hating ends, can spontaneously assemble into vesicles that encapsulate whatever solutes happen to be nearby. The importance of compartments in origin of life thinking cannot be overstated, because without some kind of boundary, any novel combination of molecules would immediately diffuse away into the vastness of its surroundings, unable to retain advantages or compete as a unit. Modern cells use lipid bilayers made of phospholipids, complex molecules with charged heads and hydrophobic tails, but simpler fatty acids, readily synthesized in prebiotic experiments or delivered by meteorites, can also form vesicles in water with an appealing property. They dynamically exchange components with their environment, growing by incorporating more fatty acids and dividing when shear forces or osmotic pressures distort them. Jack Szostak's laboratory at Harvard and later at Massachusetts General Hospital made major contributions to this field by showing that fatty acid vesicles can take up nucleotides, that polymerization of RNA inside them is possible under certain conditions, and that vesicles with internal RNA can, under gentle agitation and osmotic pressure, changes divide into daughter vesicles that inherit some of that RNA, setting the stage for a primitive form of heredity tied to compartments. Adding mineral particles like montmorillonite clay enhances nucleotide polymerization and vesicle formation, hinting that on the early Earth, muddy shorelines, not pristine glass flasks, may have been the true reaction vessels where greasy bubbles captured random assortments of polymers, some of which occasionally conferred slight advantages in stability or replication, and thus persisted across cycles of growth and fragmentation. While membranes and metabolism like reaction networks provide structure and energy, life as we know it also requires a way to store information and pass it on with variation. And for decades, proteins seemed the only plausible catalysts for biochemical reactions, whereas DNA, with its simple four-letter code and elegant double helix, looked like the obvious candidate for information storage. This division of labour posed a chicken and egg problem, because without proteins, how could DNA be replicated? And without DNA, why would proteins exist in specific sequences? But in the 1980s, an unexpected discovery reconfigured the puzzle. Thomas Tse, working on the ciliate tetrahymena, and Sidney Altman, studying bacterial RNase P, independently found that certain RNA molecules can catalyze reactions, cleaving and ligating other RNAs or themselves without any protein assistance. These catalytic RNAs, dubbed ribozymes, demonstrated that RNA can, in principle, both carry genetic information and perform enzymatic functions, leading to the proposal of an RNA world, in which, before DNA and proteins took over their current roles, networks of RNA molecules replicated, mutated, and evolved, perhaps aided by simple peptides or minerals but not yet dependent on the full complexity of modern biochemistry. Evidence supporting this idea comes from many corners. The core of the ribosome, the molecular machine that builds every protein in every known organism, is itself made of RNA, with proteins playing supporting roles, implying that RNA had a central place early in life's history, and many essential coenzymes, such as ATP or NAD, include adenosine or other nucleotide-like structures, as if they were adapted from a prior era when nucleotides did far more varied work. However, Proposing an RNA world is easier than showing how such a world could plausibly arise from simple molecules under prebiotic conditions, and early attempts to synthesize the nucleotide building blocks of RNA from likely primordial ingredients ran into roadblocks because the straightforward routes that organic chemists envisioned in flasks didn't mesh well with what geochemists knew about early Earth environments. For example, assembling a ribonucleotide requires a sugar, ribose, a nitrogenous base, and a phosphate to come together in the right orientations. But ribose itself is unstable, and in uncontrolled mixtures, tends to form tangled families of related sugars rather than a clean product. In 2009, chemist John Sutherland and colleagues published a breakthrough synthesis that cleverly bypassed the need to build ribonucleotides from fully formed ribose and free bases, instead starting from simple compounds like glycolaldehyde, cyanamide, and cyanoacetylene, which under ultraviolet light and with phosphate present, could be coaxed through a sequence of reactions that yielded activated pyrimidine ribonucleotides, the C and U of RNA, in respectable yields, with the key insight being that nature might favour reaction pathways that share intermediates and conditions, turning apparent side reactions into steps along the main route. Later work extended these principles to purine nucleotides and showed how such molecules, once formed, can undergo template-directed polymerization, where short strands of RNA guide the addition of complementary nucleotides to form longer strands, especially when aided by cycles of drying and rehydration or by subtle pH changes in microenvironments like rock pores or evaporating films. Even with nucleotides in hand, demonstrating non-enzymatic RNA replication that approaches the fidelity and speed needed for Darwinian evolution remains an ongoing challenge. Though progress has been steady, experiments show that certain activated nucleotides spontaneously assemble into short polymers on clay surfaces or within eutectic phases of ice, where freezing concentrates solutes into microscopic brine channels, and that these polymers can, under the right conditions, act as templates to direct the formation of complementary strands, albeit with frequent errors and a limited maximum length before strand separation becomes difficult. In 2009, Gerald Joyce and Tracy Lincoln evolved, within the controlled confines of a laboratory, pairs of ribozymes that can catalyze each other's synthesis from smaller RNA fragments, a kind of cross-replicating system that, while heavily engineered and dependent on supplied substrates, hints at the plausibility of an RNA-based replicator once a certain threshold of complexity is crossed. Other researchers have found ribozymes that can ligate pieces of RNA, add nucleotides non-templated, or even carry out a rudimentary form of RNA polymerase activity, extending a primer along a template, though usually only for short stretches. Each of these achievements, while still far from a fully autonomous, self-maintaining RNA world, adds another tile to the mosaic, suggesting that no fundamental chemical law forbids RNA or similar polymers from spontaneously assuming the dual role of archive and catalyst, but also underscoring how narrow the path may have been between inert soups and evolving cycles, a path that almost certainly intertwined with energy gradients, mineral surfaces, and compartmentalization rather than floating in isolation. Parallel to gene-first scenarios like the RNA world, some chemists and biochemists have championed metabolism-first models, which posit that self-sustaining networks of chemical reactions resembling primitive metabolisms might have arisen on mineral surfaces or in geochemically structured environments before the appearance of informational polymers, with heredity emerging later as a refinement of systems already cycling matter and energy. German chemist Günther Wächtershäuser was particularly influential here, imagining an iron-sulfur world in which early life began on the surfaces of pyrite, thus Fs2, and related minerals near volcanic vents, where gases like carbon monoxide, hydrogen sulfide, and ammonia reacted under the catalytic influence of transition metals like iron and nickel to form activated thioester compounds, analogous to modern metabolic intermediates, potentially driving sequences that resembled parts of the Krebs cycle used by contemporary cells to harvest energy. Wächtershäuser emphasized the importance of surface-bound intermediates, arguing that they would keep nascent reaction networks localized and shielded from dilution, and he suggested that selection could operate even on such purely chemical systems, favoring those networks that grew autocatalytic, able to use external fluxes of simple molecules to sustain and amplify their own composition. Later experiments by researchers like Eric Smith, Harold Morowitz, and Marcus Ralzer have shown that under certain conditions, metals and simple inorganic catalysts can indeed promote sequences of reactions that map onto modern central metabolism, such as segments of the reverse citric acid cycle, lending some credence to the idea that our deeply conserved biochemical pathways bear a fossil imprint of geochemical reaction networks that long predated genes. As these diverse lines of inquiry, vent chemistry, hot spring cycles, RNA catalysis, mineral-driven metabolisms, fatty acid vesicles, proliferated, the once monolithic image of a single, primordial soup giving birth to life in a moment of dramatic self-assembly gave way to a more intricate panorama of overlapping niches and gradual interlocking advances in which no single factor sufficed but multiple, mutually reinforcing processes nudged matter toward biology. One can imagine, for instance, an alkaline vent mound on the ocean floor where proton gradients drive the reduction of carbon dioxide into organic acids along mineral surfaces, generating a local stock of small metabolites. While far above, in a volcanic region, pools at a hot spring's edge experience daily evaporation that strings together nucleotides and amino acids into short polymers, some of which assemble into vesicles when storms wash them downslope. Impacts from meteorites pepper these landscapes with extraterrestrial organics and create, in their craters, transient hydrothermal systems that mix deep geochemistry with surface chemistry. Over millions of years, polymers that happen to promote their own replication or the stability of their compartments gain an edge in this complex environment, and at some point, perhaps in multiple locales, systems emerge that combine an energy-harvesting reaction network, an information-bearing polymer, and a protective membrane, giving rise to lineages that can truly evolve as units, competing and recombining as they spread into new niches, so that what we casually call the origin of life may in fact have been a distributed, iterative process, more like the gradual ignition of a forest than the flip of a single switch. While chemists and theorists reconstruct hypothetical steps, paleobiologists and geologists scour the ancient crust for constraints on timing and nature, using every available clue in rocks that have endured billions of years of metamorphism, tectonics, and erosion. One of the most evocative types of evidence comes from stromatolites, layered structures built by microbial mats that trap, bind, and sometimes precipitate sediments as they grow toward light, whose living equivalents can be seen today in places like Shark Bay, Australia, where cyanobacteria build laminated domes in hypersaline shallows, but whose fossilized versions stretch back deep into the Archean. In the Pilbara Craton of Western Australia, 343 billion-year-old sediments host conical and domal stromatolites, whose fine layering and shape strongly resemble those made by microbes, suggesting that by then complex microbial ecosystems capable of coordinated growth and mineral manipulation had already diversified across shallow marine platforms. Even older, around 3.7 billion years, claimed stromatolites in Greenland's Isua supracrustal belt have sparked intense debate, with some geologists arguing that their banded, mound-like structures are products of deformation and sedimentary processes rather than biology, while others point to subtle isotopic signatures and microtextures as evidence of microbial influence, illustrating how difficult it is to confidently interpret rocks that have been heated and squeezed multiple times. In parallel, researchers examine individual microscopic features, filamentous or spherical forms preserved in cherts, and the ratios of stable isotopes like carbon and sulfur to infer biological fractionation. Purported 3.5 billion-year-old microfossils from the apex chert of Western Australia, once celebrated as the oldest known cells, were later reinterpreted by some as abiotic mineral artifacts, reminding the field that extraordinary claims demand rigorous, multi-pronged analysis, while more recent finds in South African Barberton greenstone belt rocks show microstructures associated with ancient seafloor hydrothermal systems that may record sulfur metabolizing microbes around 3.4 billion years ago. Sulfur isotopes, particularly mass-independent fractionation patterns, reveal when atmospheric oxygen began to accumulate, because certain anomalies disappear once ozone and oxygen levels rise beyond a threshold, and these data, combined with iron formations, push the onset of significant atmospheric oxygenation, the Great Oxidation Event, to around 2.4 billion years ago, implying that for a billion years or more before that, life thrived in largely anoxic environments. Throughout these arguments, one consistent thread emerges. By 3.5 billion years ago, and probably earlier, Earth hosted diverse microbial communities exploiting different energy sources, from chemolitho, autotrophy in vents, to anoxygenic photosynthesis in shallow seas, meaning that the actual origin of life must lie significantly earlier in the rock record, during an interval from about 4.3 to 3.8 billion years ago, that is geologically shadowy but chemically crucial. Among those early microbial pioneers, the lineage that would utterly transform Earth's surface chemistry was that of the cyanobacteria, which evolved the ability to split water using sunlight, releasing molecular oxygen as a byproduct in a process called oxygenic photosynthesis. In modern ecosystems, cyanobacteria form blooms in lakes, endosymbiotic plastids in algae, and intricate mats in tidal flats. But in the Archean eon, when reducing conditions prevailed in oceans saturated with dissolved iron, their waste product was a potent oxidant that initially reacted with available iron to precipitate banded iron formations, alternating layers of iron oxides and silica that today constitute some of our richest iron ore deposits. Over time, as sinks for oxygen in the oceans and crust became saturated, free O2 began to build up in the atmosphere, oxidizing methane, cooling the climate, and possibly triggering global-scale glaciations, while simultaneously opening up vast new metabolic possibilities, including aerobic respiration with its far greater ATP yield per unit of fuel, setting the stage for more complex cellular architectures. Genetic studies suggest that photosynthetic apparatus, including the core reaction centers of photosystems I and II, evolved through gene duplication and tinkering in ancestral bacteria, and that horizontal gene transfer spread parts of this machinery across lineages, while the ultimate installation of a cyanobacterial descendant as a permanent lodger within a eukaryotic host cell gave rise to chloroplasts, the green factories in plant cells, so that every forest and prairie today is, at its deepest level, an echo of ancient microbial innovations. However, all of these evolutionary fireworks took place within a biosphere already in full swing, and to reach backward from them toward life's last universal common ancestor, scientists turned not to rocks, but to molecular sequences. In the late 1970s, microbiologist Carl Woese, working in relative obscurity in Illinois with a small group and laborious methods, compared the sequences of ribosomal RNA from a variety of organisms, reasoning that because ribosomes are essential and ancient, their RNA components should carry deep phylogenetic signals. What emerged from his painstaking cataloguing of oligonucleotide fragments was a shock. Instead of a neat tree with bacteria at the base and eukaryotes branching off later, he found that prokaryotic life split into two fundamentally distinct domains, which he called bacteria and archaea, with eukaryotes forming a third branch more closely related to some archaea than to bacteria, a three-domain model that overturned the traditional prokaryote-eukaryote dichotomy and revealed just how strange and diverse microbial life truly is. By mapping gene presence and absence and reconstructing ancestral states, researchers have attempted to infer the features of the last universal common ancestor, LUCA, not as the first living entity, but as the most recent organism from which all extant life descends, and the portrait that emerges is of a complex, already sophisticated cell, with DNA as its genetic material, RNA intermediaries, and a full translation apparatus including ribosomes, tRNAs, and a nearly modern genetic code, as well as membrane-associated energy metabolism that may have involved proton gradients and chemiosmotic machinery, perhaps situated near hydrothermal settings. Intriguingly, some reconstructions suggest that LUCA possessed enzymes adapted to environments rich in hydrogen and carbon dioxide but lacking oxygen, and that its core metabolism resembled that of modern, autotrophic archaea and bacteria inhabiting vents, lending circumstantial support to vent origin hypotheses, though these inferences remain contested and clouded by the pervasive horizontal transfer of genes among early lineages. Horizontal gene transfer, the movement of genetic material between unrelated organisms, complicates efforts to draw a simple branching tree of life back to a single trunk, especially for the earliest chapters when cell boundaries and replication mechanisms were less rigid. Microbes routinely exchange plasmids, viruses shuttle DNA between hosts, and mechanisms like transformation, and conjugation blur lineage distinctions, so that instead of a neat arboreal diagram, some biologists envision a tangled web or even a reticulate network for early evolution, with modules of genes for metabolism, information processing, and cell structure hopping between lineages in a dynamic communal pool. In this view, the genetic code's near universality across life is less a reflection of strict vertical descent from a single protocell and more a consequence of selection for compatibility in this gene-sharing milieu. Because once one coding scheme gained a foothold and spread through horizontal transfer, any deviance would be at a disadvantage when swapping genes, a scenario Francis Crick alluded to when he called the code a frozen accident that may have started as one of many possibilities but became locked in once interdependence grew. Modern comparative genomics reveals that while some parts of the code are indeed arbitrary assignments, others reflect underlying chemical affinities that minimize the impact of common mutations or translational errors, suggesting that even in this supposedly accidental structure, selection has sculpted robustness. And this interplay of contingency and necessity recurs throughout efforts to reconstruct life's beginnings, reminding us that while certain principles like energy gradients and compartmentalization may be nearly inevitable, the specific solutions evolution found, RNA versus some alternative polymer, lipid bilayers versus other boundaries, could easily have turned out differently. As origin of life research advanced, synthetic biologists began to test just how flexible the definition of life might be by engineering cells and molecules that stretch or rewrite what had long been assumed fundamental. In 2010, a team at the Craig Venter Institute announced that they had successfully created a synthetic bacterial genome, laboriously stitched together from chemically synthesized DNA fragments, and transplanted it into a closely related cell whose own DNA had been removed, effectively rebooting the recipient with a human-designed chromosome, which the team whimsically encoded with hidden watermarks including famous quotes. Later, in 2016, the same group constructed a minimal cell genome, JCVI CN 3.0, containing just 423 genes, by systematically stripping away genes from a mycoplasma strain and testing which combinations still produced a viable, self-replicating organism, revealing that nearly a third of the essential genes had unknown functions, underscoring how much we still don't understand about even the simplest modern cells. Elsewhere, researchers expanded the genetic alphabet by introducing unnatural base pairs into DNA, creating semi-synthetic organisms that can store information in 6-letter codes, and chemists synthesized alternative nucleic acid backbones dubbed XNAs that can also store and propagate genetic information, hinting that life, in principle, could operate with very different molecular substrates. These feats do not recreate the conditions of early Earth, but they illuminate the space of possible biochemistries and show that the particular choices evolution made on our planet, ribose, phosphate, ACGTU, are not the only functional configurations, raising the possibility that the true origin of life on Earth is one specific trajectory within a much larger landscape of potential living chemistries, one that we're only beginning to map. If modern synthetic cells reveal the minimum complexity for life today, studies of extremophiles, organisms thriving in what were once thought to be uninhabitable conditions, illustrate how robust once evolved life can be, highlighting by contrast how delicate the initial emergence may have been. Kilometers below the ocean floor, microbes eke out existence in sediments with vanishingly low energy fluxes, metabolizing at such slow rates that their generation times stretch into centuries, blurring the line between active life and suspended animation. In the scalding acidic pools of places like Kamchatka or Yellowstone, archaeal communities maintain proton gradients across membranes, challenged by external pH values near zero. Their proteins stabilized by specialized folds and ionic interactions. In the Chernobyl exclusion zone, certain fungi appear to use melanin as a pigment to harvest ionizing radiation for growth, while Deinococcus radiodurans survives doses of radiation that shatter its genome into dozens of fragments, reassembling it through highly efficient DNA repair pathways. These adaptations, extraordinary as they are, didn't appear overnight but were built atop pre-existing mechanisms honed over billions of years in milder settings, reminding us that while life, once established, is extraordinarily tenacious and inventive, the step from non-life to the first self-maintaining evolvable system might have required a far narrower set of circumstances, a tightrope walk over a chasm that, if crossed only once, left no direct fossils of its earliest balancing poles. In recent years, experimental technology has allowed origin of life researchers to mimic dynamic environmental conditions more faithfully, and to search for emergent behaviours in complex chemical systems without predefining all the steps, using microfluidic devices that control temperature, flow and concentration in microscopic channels, or automated platforms that mix and heat thousands of reactions in parallel, guided by algorithms that look for increases in complexity or autocatalysis. For example, some teams have built protocell reactors, where lipid vesicles, nucleotides and catalysts circulate through networks of pores and channels, experiencing oscillating pH and ionic conditions analogous to those at vent chimneys, observing how certain compositions persist or grow over cycles, while others disintegrate, offering glimpses of chemical evolution on a laboratory timescale. Computer models simulate millions of interacting reactions in abstract chemical spaces, searching for sets of molecules that mutually catalyze each other's production, forming autocatalytic networks that can grow and adapt in response to changing inputs. And while these models simplify away much messy detail, they help clarify which conceptual features, feedback loops, compartmentalization, diversity of building blocks, are needed for open-ended evolution. At the same time, planetary scientists and astrobiologists design missions to Mars, Europa and Enceladus, not merely to hunt for present-day microbes, but to reconstruct the early histories of these worlds, because finding independent life elsewhere in our solar system, or confirming its absence despite seemingly favourable conditions, would profoundly constrain the likelihood and pathways of abiogenesis, informing our understanding of how typical or exceptional Earth's story has been. On Mars, for instance, orbital observations and rover missions have revealed ancient river deltas, lake beds, and minerals like clays and sulfates that form in liquid water, along with organic molecules in drilled samples, though not yet with the complex patterns that would signal life. Europa and Enceladus, icy moons of Jupiter and Saturn, harbour subsurface oceans warmed by tidal flexing, and Enceladus, in particular, vents plumes of salty water and organic compounds into space, where instruments on the Cassini spacecraft measured hints of hydrothermal activity at the seafloor, conditions that may echo those of Earth's own vent systems in its youth. Upcoming missions such as Europa Clipper and conceptual probes to sample Enceladus's plumes aim to detect biosignatures like specific amino acid distributions, complex lipids, or isotopic anomalies, and even a null result would be instructive. If life fails to appear in multiple seemingly suitable environments, then perhaps the route from chemistry to biology requires an unusually rare confluence of factors, whereas if life turns out to be common, popping up wherever rocks, water and energy coexist, then the fragile emergence we imagine on Earth might in fact be a routine cosmic event. Either way, as we refine our experiments, peer deeper into ancient rocks, engineer novel molecular systems, and send instruments across the solar system, we layer new evidence onto the old question that first stirred in myths and philosophies. How did the silent, indifferent matter of a young planet begin to copy and transform itself in ways that would, billions of years later, produce beings capable of telling this story back to the universe and recognizing in the flowing rivers of data the fragile fossils of cells and the glowing walls of hydrothermal chimneys, the long, unfinished arc that links the first flicker of metabolism in a rock pore or hot spring to the complexity and curiosity that now seeks to understand it?